Hello, everyone, and thank you for joining us today. Today we have a guest, Clara Zemp. She's a professor of conservation biology at the University of Neuchâtel, and her research lies at the intersection between biodiversity, climate, and land use sciences, aiming to guide biodiversity conservation strategies in this changing world. She is also a co-director at of the interdisciplinary master in conservation, biodiversity conservation. She is committed to promoting a dialogue between science, politics, and society. And as part of this endeavor, she's a member of the Scientific Council of the Swiss Biodiversity Forum, which represents the National Center of Expertise in Biodiversity Research, and a member of the Young Swiss Academy, where she plays an ambassador role for inter and transdisciplinary science. She conducted her PhD studies at the Humboldt University in Berlin in geography on vegetation rainfall feedbacks in the Amazon region. She was then an associate researcher at the Potsdam Institute for Climate Impact Research and the National Institute for Space Research in Brazil. Her work provided a better understanding of the effects of deforestation and climate change on the resilience of Amazonian forests. In 2016, she joined University of Göttingen as a postdoc researcher, where she coordinated an experimental site for biodiversity enrichment in an oil palm landscape in Sumatra, Indonesia. And her work demonstrated the importance of establishing tree islands to increase species biodiversity and ecosystem services in oil palm dominated landscape. During her studies, Clara received recognition for her work. She received the Xavier Bernam Foundation Dissertation Prize from the French Academy of Agriculture and the Merian Prize from the European Society for Tropical Ecology. Her talk today will focus on the work developed almost 10 years ago in Sumatra. In particular, she will present the effect of the experiment on multiple taxonomic groups from bacteria to birds at local and landscape scale. Clara, thank you for joining us today, and the floor is yours.
Thank you so much, Ramona. Is this working well? Yeah. Okay. To change the slides, just the laptop. Okay. So hello, everyone. It's my great pleasure to be here today. My presentation will really focus on the multi-scale perspective. So I will really emphasize how do we study biodiversity change from local to landscape. So I'd like to start with an overview of this cash crop landscape that we can commonly find in Southeast Asia. In particular, here it's in Indonesia. It's an industrial scale plantation. And this typically can reach up to hundreds or thousands of hectares of monoculture, intensively managed. So as you can see, very structurally simplified, intensively managed with pesticides, herbicides, to produce as much as possible palm oil. And well, the reason for the oil palm production in this area is that it is one of the most efficient crop. So if you compare, for example, the amount of oil that you can produce with palm with the amount of oil you can produce per area, per hectare for other croplands, like, for example, rape, sunflower, or soy, this is almost 5 times as efficient. So if you would replace oil palm with other crops, you potentially would need way more surfaces. Although, of course, they do not grow in the same region. And as we know, the major problem of oil palm is that it grows where we find naturally tropical rainforests. So this is the distribution in blue of palm oil potentially, so where palm oil could grow in the world. So we recognize, of course, the major tropical regions, and where also one of the most species-rich ecosystems are found, like the Amazon forest, the forest in Congo Basin, and in Southeast Asia. So in particular, oil palm is produced in Indonesia and Malaysia. 80% of the global production is found in this region. And this is where we now focus on. So as mentioned, I was part of a project that is based in Sumatra, Indonesia. So it's this island here, in particular in the province of Jambi. And it's quite an interesting area because it's really at the frontier of forest and oil palm, between forest and oil palm, as you can see in this picture. This is one of the last rainforests that is found in this area. It's one of the last lowland rainforests left. It's a restoration concession directly adjacent to a conventional oil palm plantation. So it very illustrates the tension that exists between intensive production and natural ecosystem conservation. So a great challenge is, how do we do with this situation? Of course, from a conservation perspective, the top priority must remain conservation of intact forests. But at the same time, is it possible to mitigate the ecological damage in existing plantations? As we know that these oil palm landscapes are there and are here to stay. It will probably continue to increase in the future. So how do we manage these landscapes that are dominated by intensive oil palm monoculture? And is it possible to enrich biodiversity? So one of the ideas is to establish tree islands, so patches of trees spread in the landscape. They could have different number of species, different sizes. And the idea is that these patches should provide some habitat for some species that are able to survive or to thrive in these landscapes. At the same time, also allow connectivity for species to move in the landscape and connect more natural ecosystems. And at the same time, potentially providing some ecosystem functions and services that could directly benefit the people who work and live in these regions. But of course, we still have no idea whether this is a good idea in such cash crop landscape. So is it effective? Does it work? Does it allow to enrich biodiversity locally at the landscape scale? How to do it practically? What are the best size of the island? How many tree species do we need to plant? What is the influence of the landscape context? What distance to forest, and so on? So all these questions are really still very open. And finally, also from a scientific perspective, we are really eager to understand the mechanisms, because while we understand mechanisms, we also can better predict and understand what could occur in different contexts, for example. So to answer these questions, we established the biodiversity enrichment experiment. It's part of the largest network of tree diversity experiments worldwide. But this one was particular because it is the only one based in oil palm monoculture. So it was established more than 10 years ago in an oil palm landscape of 140 hectares dominated by monoculture. And in this landscape, we established 56 study plots, including 4 controls, so management as usual, and 52 experimental plots or tree islands. And these tree islands are shown here. You see 6 different species were planted. And this follows a gradient of planted diversity from low to high diversity, as you see with a change in color from 0 to 6. And also the size, so the area of the tree islands vary from 5 by 5 to 40 by 40 square meters. All the species are native, multi-purpose, so they are also used locally for different aspects, different products. And they were planted in intercropping with the oil palm at a 2-meter grid planting density. So here's a view from one of these islands. It's one of the largest islands, 40 times 40. And in this island, 6 different tree species were planted. This was done 5 years after establishment of the experiments. We can already see some species that managed to perform quite well, have a high survival rate, and also fast growth, reach almost the size of the old palm, meaning about 15 to 20 meters in a couple of years. So you can, for those of you, it's quite difficult on this image, but they are basically different species, 4 of which conducted pretty well. Some did not perform well in this experiment. And here is the view from the inside. So this is where we would enter to do our research. So all the students, the assistants, the researchers, during 10 years conducted research in these 3 islands. So first, very brief, some results about the structure, because this is the structure of the habitats that provides the basis for all further understanding of what is happening for biodiversity. So we use the terrestrial laser scanner that is able to capture 3-dimensional aspects of the vegetation, and we quantify structural complexity index that quantifies how complex the habitat is, from low complexity, that means very homogeneous in the vertical layers, to highly complex, that means multiple vertical layers, and also dense and compact use of space in 3 dimensions. And so what we can see here are the change in structural complexity index over time, so 2016 until 2022, and we see generally an increase in structural complexity, although it tends to stabilize after a couple of years. So basically, this planting allows to build rapidly structural complex islands, and then it tends to reach a plateau. We can also see that a couple of years after the experiments, this is already way more complex than open monoculture, as you see on the left, but also much lower compared to a natural forest. So here again, an argument mentioning that it does not replace forest, and that's also not the point here. But what is interesting is that it allows to rapidly build structural complexity in these monoculture plantations. So what are the impacts on biodiversity? We conducted sampling inventories for different groups of organisms, from soil microorganisms, fungi, bacteria, up to birds and bats. And we looked into different indicators of species diversity. And you can see here the change of species diversity for richness, or for Simpson diversity for these different groups. And we can compare the level of species diversity in the gray polygon, which is the conventional old palm plantation, to the level we found in our 3 islands in green. So overall, we see an increase for most of this taxonomy group of species diversity. Also, of course, it depends on the group, and also depends on the indicator. For example, if we account for all the species or only the most common and abundant ones, as in the Simpson diversity. So it might change. I'm not going too much into the detail. We can, of course, come back to it if you're interested. But I would like to go a step further and trying to disentangle the effects of the experimental treatment. What is the impact of tree diversity? What is the impact of the island area? So we built a structural equation model, quantifying the direct and indirect effects of this treatment on multitaxa diversity. So the intermediary part here in the figure represents the indirect relationship that are caused by changes in the ecosystem structure. And in particular, we have 2 different dimensions of the ecosystem structure. The first one is in terms of structural complexity, from low complexity, that means with a lot of gaps and little vertical stratification, to high complexity, to dense and complex structure. So this will be the first dimension. And the second dimension is more related to the presence and dominance of trees in the canopies. So trees are, as keystone structure, being different from the surrounding alpine plantations. And what we can see is that the effect of island area is significant on multi-taxa diversity, richness, or Simpson diversity, and that the effect is indirect through changes in this ecosystem structure. So the larger the island, the more multi-taxa diversity, and this is not because of a sampling effect, but because of this keystone structure that the islands represent. So now if we try to tease apart the effect of the different group of organisms, you find here the estimates in the model for the different groups. Again, Simpson and species richness. Everything that is on the right is a positive effect. Everything that is on the left is a negative effect. So we see island area has always a positive effect for all these groups. In gray, these are the indirect, so through changes in ecosystem structure. And we can see mainly, for example, arthropods and seeds, as well as trees, are positively affected indirectly by island area through changes in tree dominance. We also see some direct effects, for example, for soil fauna. Quite interestingly, the effect of number of tree species is a bit more complex. It really depends on the group of organisms, with some group benefiting the more structurally complex ecosystem, for example, the bats and understory vegetation, whereas some groups were more diverse in areas with more gaps, such as the understory arthropods and mainly the pollinators, and the seeds. So here, as you can see, of course, it depends on the organisms. It's not very surprising, but what is interesting here is really quantifying the effect of the ecosystem structure on these different groups. We also went a step further and looked into different functional aspects. So different indicators of ecosystem functioning. Also, for example, looking more specifically into predators, herbivores, decomposers, and so on. So here again, depending on the indicator, the picture varies, so there is no one-fit-all solution. But still, in general, we find that larger islands positively influence many aspects of the ecosystem functioning, being the abundance of herbivores, of decomposers, of predators. Okay, so far I gave you an overview of the response of biodiversity in the tree islands as a response of the local characteristics of these islands, the area and the number of tree species planted. But we have to keep in mind that these islands are embedded in a landscape with multiple elements in this landscape that contribute to change in biodiversity locally. And also, it's not only important biodiversity locally, but also how species composition change from one island to the other. So how is the species turnover, for example? How do species composition change from one island to the other? So to understand the influence of the landscape on local biodiversity, we looked into the landscape element in a 100-meter 100-meter cycle around the tree islands. And we looked into the presence of secondary forest patches and isolated trees that were already present in these landscapes. So you see, for example, all the yellow dots that represent isolated scattered trees that still exist. And here is basically the same structure equation model that I presented before, but now accounting for local properties in terms of the soil and this landscape elements, the number of scattered trees and the distance to forest. So this is specifically focusing on woody plants, so all the trees, lianas, and shrubs that arrive spontaneously in the tree islands. So we look at one taxa in this case. And what we can see, again, direct and indirect effects of the experimental treatment, planted diversity, island area, on the number of species of these woody plants. But also quite surprisingly, the landscape element here do not play such a big role as compared to other effects such as soil characteristics, soil nutrients, soil carbon. And this is quite consistent across the different facets of biodiversity. So if we look now into other dimensions like phylogenetic or functional diversity. So phylogenetic diversity quantifies the diversity within natural regenerated plants, but accounting for the history, evolutionary history between the relationship of the evolutionary history across species. So we see a quite consistent pattern, again, influence of the soil properties and not so much of the landscape. For the functional diversity, it's also quite interesting to see the direct and indirect effect of the experimental treatment when looking into functional traits and how diverse the communities of plants are in terms of functional traits. So now we would be interested in understanding a bit more the scale at which the landscape influences biodiversity. I showed you before an example with a 100-meter radius, but is it really good radius? How about other scales? At which scale does the landscape play an influence on biodiversity for different groups? Perhaps for trees is not the same as for bacteria. So to do that, we use the multiscale approach. So basically across, around each island, we looked at the influence of the landscape at 50-meter interval increasing radius. And for these different radius, we looked into different aspects of the landscape, accounting only for the tree islands, so more from a meta-community perspective, looking into influence of other islands on the focal patch. So looking at aspects such as the mean area of all the islands, on the density of these islands, or the distance. And we also were interested in other elements of the landscape, like, for example, orchards or secondary forests, and how the diversity of these landscape elements at increasing radius influences biodiversity. So we look into land cover richness or diversity based on also relative cover of these landscape elements. And again, distance to forest and densities of scattered trees. And not forgetting, of course, local properties that determine the treatment, how many trees were planted, and on which area. So here are the results when looking at different taxa and when looking at the scale at which the landscape influences these different taxa. So we see on the x-axis the spatial extent from local to up to 700 meters around the focal patch. And on the y-axis, we see the explained variance of the model. And you see the cycle that indicates the scale at which the explained variance is the highest. So what you can see is that, of course, it depends across taxa. And quite surprisingly, for above and below ground diversity, the landscape has a big influence, especially at large spatial scales. For example, you can see the soil fauna that is influenced at scales such as 300 up to 500 meters. Also, soil fungi, this is quite unexpected because we can naively assume that for small organisms that perhaps have a lower dispersal capacity than larger organisms like birds, for example, that the landscape element is less of an influence. But we actually see that even from macroorganisms, bacteria, and soil fungi, the landscape has a very strong impact on diversity. And this, regardless of the diversity indicator that we use, being species richness, Shannon, or Simpson diversity. Of course, it depends on the indicator, but overall, the landscape has an influence on these different indicators. Okay, so far I was talking about the influence of the landscape on local diversity. By local diversity, I mean alpha diversity. So the diversity that we find within the habitat, within the islands. Now, if we look in the landscape, we can also look at other scales of biodiversity, in particular gamma diversity, which is the total diversity we find in the landscape, and beta diversity, which is the differences or the differentiation between or across the islands. So the higher beta diversity, the more differentiation there are between species communities across islands. And this beta diversity can be decomposed in two components, the turnover and nestedness. Nestedness means that the species composition that you have in one island is a subset of the composition you have in another island. So the composition is not intrinsically different, but it's just a subset. Where turnover indicates that the species composition you find in one island differ from the species composition in another island. So there is a turnover in space in the species composition. And here, for each group of organisms, we can quantify these different dimensions of biodiversity, alpha, beta, and gamma, and we can decompose beta diversity into these two components, turnover and nestedness. So this is what we did here, looking into beta diversity, so the differentiation across 3 islands for these different groups of organisms again, and trying to see which beta diversity is related to turnover or to nestedness. And what is quite interesting is for most groups, particular for arthropods, for understory vegetation and soil fauna and soil microorganisms, the most important aspect or the highest share is represented by turnover. So that means each species composition differ from one island to the next and is not just a subset of another island. So that means in a certain way, all the habitat, all the 3 islands contain fundamentally different species from one island to the other. And in a way, it's kind of a fragmentation effect, if we wish, that each island is disconnected and contains its own and unique species composition. So from a landscape perspective, that's quite interesting because you have a higher total diversity in the end. You have a higher gamma diversity if each island contains different species composition than if there would be just a subset. And then we wanted to understand a bit more the connections across these different groups. Are there any reasons why there is such a turnover across species groups in these islands? What is the interconnections across the different group of organisms? And is there any links that we could potentially interpret as biotic interactions? So we use the complex network approach. that was developed by Ullmann, that basically represents here the different group of organisms as nodes in the network. So each node represents here the turnover. And we also include information about the environmental condition, like soil heterogeneity, in this case, the phosphorus content, because we know tropical areas are limited in phosphorus, so that might be an important factor to consider. And the habitat structure, in terms of structural complexity, which is represented here on the top. And so each link between these different nodes represents a significant correlation across these different nodes. So every link that you see in this network, in this graph here, indicates a significant correlation, partial correlation in the data. So that means potentially underlying some biotic interactions. And quite interestingly is that, for example, soil fauna is quite central in this network. It's connected to the vegetation, but also to bacteria and to soil heterogeneity in phosphorus. Similarly, soil bacteria is also quite central and connected to other groups. So this highlights the importance of soil organisms in this network, which we didn't expect. We expected more like above ground organisms to play an important role, such as plants, trees. Plants do play a role, but soil fauna is really something we didn't expect, also microorganisms in particular. So some of the key findings from the last 10 years of research, in particular focusing on this cross-scale perspective. We do see, after a couple of years only, an increase in the structural complexity in these islands. And this tends to stabilize, so it's relatively rapidly building, and then it tends to stabilize. As a consequence, we see an increase in multi-taxa diversity from soil bacteria to mammals at local scale that are driven by this change in the structure, but also at landscape scale, by each island having unique species composition across these different groups. So landscape heterogeneity here is an important role in driving landscape scale multi-taxa diversity enrichment. We also show that the islands that are more diverse and that are larger increase the diversity of plants, and in particular, not only number of species, but also looking into different dimensions like phylogenetic or functional diversity. And we found that the effect of the island area is not just direct due to a sampling effect. The larger, the more species. This is a well-known relationship in biogeography. But we found that there are some mechanisms that go beyond the sampling effect, and that larger islands provide keystone structure because they are dominated by trees in the canopies, which provide resources and structure for these organisms. And finally, I showed some interconnections in different groups, in particular the importance of soil fauna, of soil biota, like including bacteria and fungi, linking the different group of organisms above and below ground at the landscape scale. So here again, landscape heterogeneity in terms of how different the islands are is important if we want to increase biodiversity at the landscape level. And finally, because that was also one of the aim that I mentioned in the beginning, is a bit of a better understanding of the mechanistic insights of biodiversity enrichment in this type of landscape. So this is a schematic figure trying to disentangle the different mechanisms from the individual to the meta-community level. And you see here the different aspects that influence the mechanisms across these different scales. From individual scale, these are mainly aspects related to survival, reproduction, and growth of organisms in terms of fitness. which we know are influenced by life history traits, in particular, their dispersal capacity, but also, of course, the breadth of the habitat. Are they specialists? Are they generalists? I didn't talk about too much about that in the talk, but we also have some results if you're interested in specific traits and characteristics of the species that influence their success. At higher level, across biological organization, we know that landscape features, that landscape connectivity is important in explaining biodiversity enrichment by connecting tree islands through long-distance dispersal and influencing the species composition that we find in these islands. And finally, accounting for biotic interactions help to understand how biodiversity changes in the landscape, not just looking into one trophic level, like only plants, for example, but also accounting for interactions with soil microorganisms, like soil bacteria, soil fungi, soil fauna. And we have provided evidence that it influences multi-taxa diversity in the landscape. Of course, there are also other aspects that are a bit less difficult to have an influence on, like environmental and environmental and demographic stochasticity. These are aspects that we do not really control. But basically for other aspects, like how the environment filters the species, these are things on which we can play on which we can influence by planting these tree islands and by recovering ecosystem structural complexity, for example. So with that, I will end my talk here. Of course, this is a large teamwork. We're more than 40 people involved, many students, many field assistants, but of course, strong collaboration with other professors, in particular, Holger Kreft and Dirk Hölscher at the University of Göttingen, as well as in Indonesia, Bang Irawan and Leti Sudnawati. And we can see the different persons who played a major role in these papers that I presented: Natalie Guerrero Ramirez, Denver Catellano, Vanessa Sanchez, and Gustavo Paterno. And yeah, with that, thank you very much for your attention.
Thank you very much, Clara. The floor is now open for questions, with the kind request to please use the microphone for the main question, and if there is a follow-up question, also for that. Nadia will help me with questions if there are some online. I think we also had some people that joined online. So.
Thank you very much for the exciting talk. I would assume that I mean the species in the islands, they very much depend on a source population, right? So the distance to the forest should be very relevant, but in your analysis, the distance to the forest did not play a major role. And at the end, also in your mechanistic framework, you pointed out the role of dispersal and immigration and so on, colonization. Mm-hmm. So how can you explain that?
Mm-hmm. Yeah, that's a very good point. So what we quantified was the forest patches that remain in this landscape, which are mainly secondary forests. And we only looked into landscape up to 700 meters. The picture that I showed in the very beginning, one of these large remaining forests, which is a larger patch and is more intact, the most intact we have in these landscapes, is further away, several kilometers away. We don't know the influence of this remaining habitats, which we expect to provide a major source of species in the region. So that is still something that we need to understand better. And indeed, so for the moment, we do not have an understanding of that. It's also quite surprising, as you say, that the distance to this secondary forest was not significant in our case. And yeah, I think it's perhaps because we look into the broader taxonomy groups and we have to go deeper into the specific functional groups, or even those organisms that are long dispersed and those that are that have different dispersal strategies. So those species that are dispersed by animals or species that are dispersed by winds might differ. So yeah, I think there is way more research to be done necessarily to disentangle the mechanistics, and we are also a bit limited with this experiment because it was not made for looking into the influence of the forest. So actually the initial idea was to look into different distance from forest, but then because of some also logistical constraints and practical aspects, like alpine managers willing to collaborate with scientists, this was not part of the final design, but it would be super interesting to know, for example, how the species diversity recovers when increasing distance from this remaining lowland rainforest that still provide large patch of habitats and species pool. So yeah, I think there is room for more experiments and more research on that. But yeah, thanks for pointing.
Thank you.
Thank you very much for your talk. I was wondering during your presentation about the ideal situations to increase this biodiversity throughout the landscape, whether it's to create these large tree islands distant of certain meters, like you show these graphs where for the fungi it's up to 500 meters, for bacteria 3 or 200 meters. They maximize the connectivity or the turnover of the species, right? Or to directly transplant inocula from primary forest or secondary forest to these three islands. What would be your thought about that?
So can we maybe
either to increase the network by creating new tree islands distant of like 500 meters to maximize the network or the connectivity, or to directly transplant ourselves inocula from primary or secondary forest, like for fungal or for sound fauna that cannot disperse that well, except by demographic processes as you showed.
Yeah, that's a very interesting question. It's a bit difficult to answer without knowing the dispersal capacity of the different organisms, and this is something we do not know, especially for like fungi or bacteria. At least I'm not aware of how can we estimate the maximum dispersal capacity of these organisms. So first, I would say we definitely need to better understand the maximal dispersal capacity, and then once we know that, we can adjust the tree island distance to reach this capacity, and also to connect as stepping stones remaining habitats. And of course, if we are further away than this maximal dispersal capacity, then we probably need to use other ways, like you mentioned, to bring by ourselves the species if they are not able to disperse by themselves. So this will be for soil microorganisms. For larger groups, I think there again is a matter of the dispersal capacity in terms of their trades, but also how able they are to move in this disturbed landscape. If I take an extreme example, which I didn't present here, of the orangutan, we know that it's actually able to move across very large distances, and also to use, to some extent, alpine landscape for looking for resources and basically using this habitat as well, if they do not have further remaining habitats in sufficient amount. So we could imagine that by having this tree island spread in the landscape, it might facilitate the movement of these large groups of the large species. But yeah, I think it's very dependent on the dispersal capacity and how able they are to move in this harsh environment condition. Because in the alpine plantation is way hotter and also less humid than in a forest. And so crossing these areas is very harsh for many species that are not adapted to, that are forest dependent. So by having these 3 islands, potentially we help providing conditions more favorable for movements of these organisms. But again, it depends on the species, their traits, and their capacity to adapt, I would say. Yeah.
Thank you.
Thank you very much for this nice presentation. In particular, the aerial photographs were very impressive, and they made it obvious that the green biomass is completely differently distributed between the monoculture and the tree island. And this made me thinking how sampling really took place, sampling of the different taxa, only from the ground, or did you combine it with canopy sampling?
Sorry, what results are you specifically referring to? I'm not sure I understood your first part of the question.
So the green biomass is differently distributed between the monoculture and the tree island.
Biomass?
The biomass, the green biomass.
I did not necessarily present biomass, but
no, but just in the system. In the sites,
okay,
in the sampling sites.
Yeah.
So in the oil palm, it's completely concentrated in the canopy, and in the tree island, it's more distributed along the vertical axis, and also in the ground. So this made me just thinking about sampling techniques. How did sampling take place? Only from the ground or in combination with canopy sampling?
So first element to answer, all the sampling were the same across all the sites. Of course, otherwise it will be difficult to compare. So this, in terms of understory arthropods, mainly pollinators, it was a sweep netting that was conducted in all the plots in the same way. We do not have canopy arthropods in the data, so the arthropods is actually misleading. It's understory arthropods, in particular pollinators. For the soil organisms, again, was the same everywhere. So sampling soil in specific subplots, like randomly spread in the islands, and then with meta-barcoding, looking into OTU diversity for fungi and for bacteria. For soil organisms, it was through also soil sampling and then extracting the organisms with a heat extractor. And then in the lab, by morphological characteristics, disentangling to morphospecies or even higher taxonomic level. So that's also something I didn't mention, that of course we do not have the same resolution for each group, depending on the skills that are present, depending on the availability of taxonomists who are able to identify species and also the resources we have. So for soil fauna, it was a way higher taxonomic level, mainly genera or family, as compared to plants, for which we had a botanist able to identify to the species level. So yeah, this differ across the groups, and I can imagine that it has an influence also on the pattern that we observe, because the more resolution we have, also the fine scale aspect might potentially also be more relevant. So yeah. But to answer your question, the sampling was the same everywhere. And I think it's also quite interesting that you mentioned that, because some study show changes in metabolism, so energy flux in the canopy and in the soil and the understory. And indeed, there is a change from forest to alpine. That in particular, in alpine, I think it was mainly dominated by below ground, like earthworm, whereas in the canopy in the forest, the energy flux is mainly dominated in the canopy and also in the soil, but with other functional groups than just earthworm. So this is something we didn't look into, but I think will be super interesting to know more, like across these different strata, canopy, or soil arthropods, for example. How does this changes? And we should see some influence also on the level in the vertical stratification where these organisms live. Yeah, it would be very interesting. Thanks.
Thanks, Clara, for the presentation. I mean, for me, the oil palm plantation is still something unbelievable. I mean, it shouldn't be allowed. But now, showing your results, it seems like possible the combination of the two things. Mm-hmm. I mean, I might be a bit provocative, but I mean, if you have a desert and you plant plants, of course, I mean, you will have like more diversity compared to the desert. And for me, the desert was like the whole plantations. And now you played around with patch size, patch structures, and so on and so forth. But does it now allow people to say, okay, but maybe planting oil is not so bad if we can, I mean, what is the comparison? Is it the forest, which I only saw once? Is it different restoration patterns to compare? I mean, what is your point there?
Yeah, thanks a lot. So yeah, that's of course an aspect we are or at least I'm very afraid of, is a misunderstanding of our results. And that's why we always repeat constantly that this does not replace conservation of intact forest. The only time where I compared with tropical forests was in terms of structure. So we look at structural complexity, and there we can see it's intermediary between both. But for the biodiversity study, we just compare with monoculture, and we see how more diverse the community is in our islands compared to conventional monoculture that we call control. Of course, if we would do the same with the rainforest, we would see first different level of diversity and also different composition. We can look into which species is forest dependent, and that's where it becomes interesting. If, for example, I didn't show it here, but in the study by Gustavo Paterno here in Science last year, he looked into the plants, the woody plants in particular, those that are forest dependent and those that are more found in open habitats or in disturbed environment. And yeah, of course, the organisms respond differently depending on this habitat. Their guilds basically where they are found in. So if we want to compare with forest, we have to look into which species that are forest dependent are able to move and able to thrive in this oil palm monoculture landscapes. So that could be something to look into further, like more specifically which species. And again, I will point to that paper. But all what I presented here was more a comparison with basically doing nothing. So what if we just enrich oil palm with biodiversity with 3 islands compared to if we don't? Now, if we want to make further recommendations, but I would not necessarily go into this direction because we know that intact forest contains species that are found only there and that will never be able to thrive in this landscape. So we could potentially continue research on which species are able to use these landscapes. But yeah, I think at least for now, it was mainly the purpose of looking at the comparing with conventional oil palm. Does that answer your question?
Is there a question? That would be the last question.
Okay, what's the yield of oil production in these 3 islands as compared to the non-3 islands? And did you find according to the different canopy density, did you find any, acceptable loss of yield? What's your guess on that?
Yeah, thanks for pointing out. I didn't mention that. So as expected, we see a loss of oil palm yield per area in the islands, also because we cut, I didn't mention, but we cut 40% of the oil palms in the islands. So less alpine, less yield, obviously per area. But what is quite interesting is that we found that this loss is in fact compensated by a gain in yield of the palms adjacent to the island. So we looked into the position 1, 2, and 3 around the island, and we found an increase in the yield. And this was quite consistent over the years. And accounting for this positive effect around the island, in the end, in the landscape, we do not have any loss or any gain, at least for now. It might change in the future because we know the competition will continue and potentially will finally decline. And that's another question, how much decline are we allowing, or the farmers obviously will not want to establish such tree islands if they know that the revenue will decline. So then it will potentially need to be compensated, or yeah, that's also very important. But for the moment, at least after a couple of years, there is no decline in the landscape.
Thank you, Clara. I'm sure there are further questions. And for this, I would like to invite those that are interested to join us for lunch. There's a cafeteria. At the cafeteria, there's a table reserved for this. Well, having Clara today here with us was a joint effort from the Biodiversity Center and the Landscape Center. So I would like to thank Nadia, Sylvia, and Catherine for this. Thank you very much, Clara, and thank you all for the interesting discussions.
Thank you.